Our next speaker is Binbin Chen. Binbin is a MD Ph.D. student at Stanford, and he'll be discussing identifying COVID-19 vaccine candidates with machine learning.
Thank you. My name is Binbin. I'm a recent PhD graduate from Ross Altman's lab. My previous research work focused on using machine learning to understand human immune system respond to cancer as well as vaccines. Thanks for giving me this opportunity to give this talk about our computation pipeline to identify vaccine candidates for COVID-19. Most of vaccines are dead pathogens or components of pathogens we can use to train immune system to get ready for real infection. We consider vaccine is one of the most powerful weapons against a pandemic like this. Not long ago, in 1950s, millions of Americans suffered from measles. Introduction of measles vaccines drastically reduced the incidence of measles in the US. Unfortunately, no vaccines are available for any form of coronavirus today. The key question we want to answer is that, what do we put into a COVID-19 vaccine? The short answer is components of viruses trigger immune responses. However, the field still has limited immunology data on COVID-19. For today's talk, I will show you by combining immunology principles and the machine learning tools trained on a wide range of known pathogens, we can predict immunogenic components of a virus. How do our immune system fight off a viral infection? First, B cell can produce antibodies binding directly to specific parts of a viral protein. It can potentially stop entry of viruses into human cells. We call parts of viral protein to be targeted by a human antibody B cell epitopes. However, what if viruses have already entered a human cell? Fortunately, human cells have machineries called MHC to present fragments of viral protein on their surface. T cells can recognize those viral protein fragments on a cell surface and destroy infected cells to stop viral infection. We call a viral protein fragment presented and recognized by T cell epitopes. Ideal COVID-19 vaccines should contain both B cell and T cell epitopes. Researchers, including our lab, have developed computational tools to identify these regions. This epitope is a linear regression model to predict where antibody binds based on structure of a target protein. Maria, NetMST Pen are two neural networks trained on a broad range of pathogen data and as well as human data to identify the region can be presented by human cells based on their sequence features. So we can run these computational tools on the whole genome of novel coronavirus. The viral genome is a single RNA producing all pathogenic protein, including spike protein. You'll hear me talk a lot about spike protein today because it enables the entry of virus into human lung cells. Human ACE2 cells are protein present in human lung cells and tightly bind to viral spike protein. This interaction is essential for the virus to enter human cells. An antibody targeting this interaction will have desirable outcome for a COVID-19 vaccine. And I show the human ACE2 protein in yellow here. We first ran this epitope on the whole spike protein. The red region indicates the likely binding sites of human antibodies. We predicted the two top epitopes near residue 440 and 494. We ran the exact same computational pipeline using a structure solved by an independent lab and to predict the same epitopes. Further, this antibody binding site predicted sits between human ACE2 and the spike protein interface. So binding to this site is likely to block viral entries. Last week, researchers in Shanghai validated our prediction using antibody isolated from COVID-19 patients. We did similar analysis for T cell epitopes of viral protein with our pre-trained neural network, Maria and the NetMST Pen. Blue colors indicates how likely these protein fragment can be presented by human cells. What we want to use for vaccines are regions with consistent dark blue colors. For example, here I show you this 9 amino acid long fragment can be presented by 65% of common MHC on the cell surface. You can find more details in our manuscript online, and we show our computational methods works well for historical SARS data. The last table in our manuscript summarizes our top candidates for potential COVID-19 vaccines. 5 out of 13 candidates are from spike protein. Some of our candidates not only look like SARS fragments, but also trigger antibody production in the previous SARS studies. In conclusion, spike protein contain both T cell and B cell epitopes. Anti-spike antibodies from patients likely block the viral entry. There are at least 4 spike protein-centered vaccine pipelines are under development. We will monitor closely for the clinical outcome of these trials. For researchers who want to go to beyond spike protein, we provide a comprehensive list of candidates for either vaccine development or understanding T cell response in COVID-19 patients. With that, I would like to thank all of your audience again, my team, my collaborators, and my funding source.
Thank you very much, Binbin. We are all very much looking forward to the arrival of the vaccines, and we are grateful for the efforts of you and everybody else working on that.